Hi guys, we have started with the proteins, and in the previous lecture we have seen the structures of the amino acid and how the peptide bonds link together to form the simple proteins. So today we will be starting with transamination and deamination type of the reactions, and these are important type of the reactions for protein metabolism process. So, what is the importance of the proteins in our body? As we have discussed in the previous lecture, that protein provide the amino acid for the protein synthesis, nitrogen atoms for nitrogen containing compounds. Energy is also provided by these amino acid and the proteins, especially when the carbohydrates and lipid resources are not available in the body, that is providing the energy. So there are so many functions of protein in the body. So body proteins that convert into the amino acid and the amino acid that convert into the complex body proteins. These amino acid when undergoes for metabolic process that is actually converting into carbon skeleton and ammonia. So the fate of this carbon skeleton will be different. The fate of the ammonia will be different. That ammonia will turns into the urea. This is more toxic ammonia. So that will be converting into urea. We will be discussing in next lecture how this ammonia is converting into the urea through the urea cycle, because this is more toxic, this is less toxic, and that have to remove from the body, right? So these amino acid that is converting in two component, carbon skeleton and ammonia, and this carbon skeleton will go for the energy and will have several fates. This carbon skeleton of the amino acid that will be either converting into pyruvate. This carbon skeleton have another fate that can be converted into acetyl CoA, pyruvate. That carbon skeleton can be converted into pyruvate, that can be converted into acetyl CoA, or that can directly goes into citric acid cycle. You have discussed and learned this citric acid cycle in previous lectures. So amino acid have three fates. The carbon skeleton of the amino acid will have three fates that can convert into acetyl CO. Again, that will going for energy that can directly goes into the citric acid cycle. And you know that citric acid cycle that is providing the energy that may go into pyruvate, and pyruvate is also providing the inner energy. Then if this carbon skeleton that is converting into acetyl CoA that have further two fates that can be converted into fatty acids, or it may be converting into ketone ketone bodies that may also provide the energy, fatty acid that is also providing the energy. And this pyruvate, as you know, that is used for making the glucose as well through the gluconeogenesis process. Pyruvate that is converting into the glucose, right? So dietary protein that we are eating that is converting into the amino acid that is making several nitrogen substances, amino alcohol hormones, hemopurines, pyrimidines, neurotransmitters, nucleotide. All of these are the nitrogen substances made by the amino acid. This is anabolic process in most of the cases, right? So this all we have discussed, that is the catabolic process. Most of the item that is made here, that is the anabolic process. So amino acid that contribute toward the anabolic process as well as the catabolic process. In the catabolic process, most often that is going for the energy type of the reactions. So the amino acid in the protein we are eating, taking from the diets, that is converting, that is storing into amino acid pools. So you can see here this dietary proteins that is converting into amino acid pools that store in your body. And from this amino acid pool that goes for glucose, glycogen formation, lipids, as we told you in the previous slide, that if acetyl CoA formed from the amino acid, that can convert it into lipids, right? And if that is converting into pyruvate, that can be goes for formation of glucose through gluconeogenesis process. So some of the non-protein compounds, heme hydrocyclic amines, we have discussed in previous slide. And in the catabolic process, when these amino acid goes for a catabolic process, that is actually producing carbon dioxide, water, urea, and energy. These amino acid from the amino acid pool that will make for new amino acids as well. Tissue proteins also produced from the amino acid pool. Enzymes, hormones, antibodies, hemoglobin. So all of these are producing from the amino acid pool. So for all of these reactions we have discussed in previous slides, there are two processes that are very important. One is the transamination reaction and other is the deamination reaction. So we will try to understand all these simple type of the reactions. So let's say this is the alanine. As you know that alanine is an amino acid. It's a simple amino acid, COH2, NH3, CH, CH3, alanine, right? When that combines with alpha ketoglutarate, what is alpha ketoglutarate? You have talked about in the Krebs cycle. If you remember, you have talked about and make the structure in the Krebs cycle. So alanine, when combined with alpha ketoglutarate, that is converting into pyruvates. As we see in the previous slides, that some of the amino acid carbon skeleton that is converting into pyruvate. So this is a kind of the amino acid, that is alanine is a kind of, if alanine is the amino acid, carbon skeleton of the alanine that will be getting separated, NH3 ammonia that will be separated, and this carbon skeleton of the alanine that will be converting into pyruvate, right? So when how this is converting into pyruvate? Through the transamination process. How this carbon skeleton that is converting into pyruvate? Through the transamination process. So alanine react with alpha ketoglutarate and the enzyme alanine amino transferase that belong to the classes of the transferase. We have seen the classes of the amino acids, enzymes. We have seen the classes of the enzymes. What is the function of the transferase? That is transferring a group. So alanine aminotransferase, that is transferring a group, right? This is the function of the aminotransferase. So this aminotransferase is performing the function of transamination. So in this reaction of alanine plus alpha ketoglutarate, aminotransferase that is producing pyruvate and glutamate. Pyruvate and glutamate, how that is doing the transfer? Basically, these are the two substances that are interchangeably transferring on each other. This is interchangeably. So you can see this O that will go here at this part, and this NH3 that will go at this part. So look at this one. This O that comes at this point, CH3CH, C double bond O. So CH3C double bond O, this O that comes at this point, Pyruvate, making the pyruvate. This is O, right? And this NH3 that will go at this point. This one you can see here. So the alpha ketoglutarate with removal of this one, and in place of this is the NH3 that will be converting into the glutamate. So what is the function of this aminotransferase? That is transferring this NH3 at this point and this O at this point, right? This is a function of the aminotransferase. So this is the whole transamination reaction, amino acid that is reacting with the alpha ketoglutarate, and aminotransferase that is shifting NH3 from the amino acid and O from the alpha ketoglutarate. So glutamate is formed. So look at this one reaction. Okay, how alpha ketoglutarate that through the transamination process, that is converting into the glutamic acid. Basically, this O will be converting into NH2, this NH2, right? So it's a transamination process, and if you have the glutamic acid, that will be converting into alpha ketoglutarate. So you can see it's a complete cycle. So when alpha ketoglutarate converting into glutamic acid through the transamination process, keto acid is also formed. And reverse of that, when glutamic acid is converting into alpha ketoglutarate, there is a synthesis of any amino acid, and you know that when glutamic acid is converting into alpha glutaric acid, this NH2 part that is removed, and that is very important for the urea cycle. So this nitrogen base is separated. This nitrogen, this nitrogen, this is separated. So it's a reversible type of the transamination, either the alpha ketoglutarate that is converting into glutamic acid, that is an amino acid, and during that process a keto acid is formed. When amino acid is converted into alpha ketoglutarate through the transamination process, that will result in the formation of alpha ketoglutarate and synthesis of any other amino acid, and in this case nitrogen NH2, nitrogen base that is getting separated. So this is the brief about transamination process. Transamination process. In the last lecture, we have talked about that there are 20 amino acids, 9 are essential amino acids, and in previous slides, we have seen that amino acid have different fates. Amino acid that can be converted into pyruvic acid, either or amino acid have a fate to convert into acetyl CoA, or amino acid that may undergo into citric acid cycle. We have seen three fates. We have seen three fates in the previous slides. You can see here again, I can show you that there are three fates. This carbon skeleton of the amino acid that can be converted into pyruvates, that can be converted into acetyl CoA, that can directly goes into citric acid cycle, right? These are the three fate: pyruvate, acetyl CoA, and citric acid cycle. So what kind of the amino acid will go in these three fates? So alanine, you have to remember all of this. Alanine, glycine, cysteine, serine, that will, what is the fate of these amino acids? That will make the pyruvic acid, right? Isoleucine, leucine, tryptophan, that may form acetyl CoA, and further acetyl CoA that is interconvertible and goes into the mitochondria. Aspartic acid and asparagine that will directly go into citric acid cycle, but at different points. You know that citric acid cycle that have different substances. So aspartic acid and asparagine that will go to form the oxaloacetic acid. Oxaloacetic acid, you know, is the part of the citric acid cycle. Other amino acid, tyrosine, phenylalanine, aspartic acid, that will also go into the citric acid cycle, but they will be forming the fumaric acid. Isoleucine, methionine, threonine, valine, that also go into the citric acid cycle, but forming the succinyl CoA. Glutamic acid, glutamine, histidine, proline, arginine, that will also go into the citric acid cycle, but where they will enter, they will be forming alpha ketoglutarate acid. As I told you about the glutamic acid example, and the aminotransferase, that will be the enzyme that is converting through the transamination process, that will be converting through the transamination process. So all of these reaction that is happening with the transamination process. We have seen the glutamic acid example that is converting into alpha ketoglutarate. Alpha ketoglutarate. We have seen here glutamic acid that is converting, this glutamic acid that is converting into alpha ketoglutarate acid. So these are the fates of different amino acids. This table also tells you about the amino acid alanine when react with alpha ketoglutarate in the transamination process, converting in the to the pyruvate plus glutamate. Aspartate reacting with the alpha ketoglutarate, converting into oxaloacetate and glutamate. So you can see glutamate is the same here, right? And here the alpha ketoglutarate is the same in these reactions. And the coenzyme that is working with these transferases, transaminases, these transaminases belong to the class of transferases, as I told you in previous slide. So the coenzyme or the prosthetic group, that is the vitamin B6. pyroxyl phosphate. That was the transamination process and how the enzyme that is working in the transamination process. Now we are talking about oxidative deamination process. Oxidative deamination process means the dehydrogenase class of the enzymes that will be using enzymes from this class that will be using, and one of the important member of the oxidoreductase class, that is the dehydrogenases, that is the important member of the oxidoreductase class. So talking about the oxidative deamination process, that is removing the amino group as an ammonium ion from the glutamate and provides alpha ketoglutarate for transamination process. We have talked about the transamination process, right? So dehydrogenase, what is the function of the dehydrogenase? We have seen in the previous lecture during the mid course, that is doing the oxidation reduction process. Oxidation reduction process, right? So look at this one. The purpose is to remove this ammonium ion. As you know, this ammonium ion that is highly toxic and that have to be converted into the urea that is comparatively less toxic. So that will have to go into the urea cycle. And this is the carbon skeleton. This remaining part from this to this part, alpha ketoglutarate, it's a carbon skeleton. So as I told you in previous slide, that the amino acid is converted into carbon skeleton and ammonia, and that ammonia that will go into the urea cycle, right? This one, the amino acids that is converted into carbon skeleton and ammonia. So how that is converted, this amino acid, how that will be converted into carbon skeleton ammonia? Through the deamination process. So look at this one. Actually, during that, it's an oxidoreductase process. So this is going to remove. This is going to remove, right? And when the valency is free, basically the oxygen, it's an oxidative deamination process. It's an oxidative deamination process. So in place of this one, oxygen will be there, right? So this will be going to remove, and in place of this one, oxygen will be taking place of that, right? So all of that structure, that will be the same glutamate structure, OOCCH, CH2, CH2, COO, OOCC, CH2, CH2, COO. All of the structure is the same, only this oxygen, this oxygen, this oxygen will be there in place of this NH3. And the enzyme that will be dehydrogenase. Glutamate in this case, we are talking about the glutamate. So the enzyme, we are talking about the glutamate. So enzyme will be glutamate dehydrogenase process. So it's an oxidative deamination. What is the basic purpose of oxidative deamination? To remove the ammonium ions. To remove the ammonium ions that is more toxic. So look this one example of deamination, oxidative deamination process. We have to remove the ammonium ions. right? So look at this one. This is the glutamic acid. It's an amino acid, you know. It's an amino acid, right? So this have to be removed. This have to be removed. So you can see when that is getting removed, this is taking, it's an oxidative process. So that is taking the place of oxygen. That is NH2 is removed and that is taking the place of oxygen, right? So this is the taking the place of oxygen. This has to be removed. So in this reaction, this, as you know, that oxidative processes, that is energy intensive process. So NAD is converting into NADH2, right? So glutamic acid will be turned into alpha ketoglutarate acid, right? Through the oxidative deamination process. And in this process, there is a formation of an amino acid. So that's all for today. Thank you very much.